O Poly, interferon gamma, and others. So we choose these hits for our study, and HLA and FA, of course, and these cytokines, we hope allow us to determine the main genetic individual characteristics of the immune system, which important in multiple myeloma.
Any more questions? If not, thank you very much. And then we proceed with the next presentation, which will be given by Anna.
Good morning, everyone. I'm Anna, and the topic of my presentation is HLA-PW4 gene combinations influence susceptibility to B-cell chronic lymphocytic leukemia. Sorry. As a background, B-cell chronic lymphocytic leukemia patients have several humoral and cellular immune abnormalities. The immune survival of tumors is mediated by recognition of antigens presented in context of HLA class 1. Both by cytotoxic T cells via T cell receptors and by natural killer cells via killer cell immunoglobulin-like receptors. Here. We have just heard many information about the cure and the ligand, but I would like to say something. Cure with two immunoglobulin-like domains interact with specific HLA-C alleles, with one exception for KIR2D4, which bound to non-classical HLA gene. The specificity of HLA-C molecules depends on presence of amino acid at the position 77 and 80. And the HLA-C group 1 molecules have a serine at the position 77 and asparagine at the position 80. And serve as ligand for KIR2D2, KIR2DS3, but if it acts as a ligand for KIR2DS2, as I just heard, it's controversial. The HLA-C group 2 molecules, which has asparagine at the position 77 and lysine at the position 80, act as ligand for KIR2D1 and 2DS1. The KIR2DS4 is postulated to recognize specifically HLA-CW4 molecules. Three domain KIR3DL1 and maybe KIR3DS1 bind to HLA-PW4. The affinity of KIR3DL1 to HLA-PW4 molecule depends on the amino acid at the position 80 in HLA-PW4, and HLA-PW4 with isoleucine at the position 80 generally exhibits stronger inhibition than those with threonine at this position. Another receptor and ligand interaction is KIR3DL2 with HLA-A3 and A11. In this presentation, we focused our attention on KIR3DL1 and KIR3DS1 and their ligand HLA-PW4. The aim of our study was to determine the association between polymorphism of KIR genes and HLA-PW4 allotypes and susceptibility to BCLL. 185 individuals with BCLL and 200 healthy subjects from the same region were enrolled in this study. The genotyping of KIR3DL1 and KIR3DS1 genes was performed by PCR SSP method. KIR ligand genes, HLA-PW4 with threonine or isoleucine at the position 80, were typed using oral SSP typing kits. Now I would like to present you our result. As you can see, the frequency of KIR3DS1 and KIR3DL1 genes did not differ in both study groups. However, we noticed that the frequency of HLA-BW4 specificity was significantly reduced in BCL patients compared with the control. Moreover, there were fewer individuals possessing HLA-BW4 with threonine at the position 80, especially those having HLA-BW4 with threonine at the position 80 while lacking HLA-BW4 with isoleucine at the position 80. Next, we analyzed the presence of KIR genes with their specific ligand genes, and we found that KIR3DS1 HLA-BW4 and KIR3DL1 HLA-BW4 genes combination were less common in BCL patients compared with the control, particularly those with the coexistence of KIR3DL1, KIR3DS1, and HLA-BW4. Among them, the frequency of individuals possessing HLA-BW4 with isoleucine at the position 80 and lacking HLA-BW4 with threonine at the position 80 was significantly reduced in BCL patients compared to the controls. As a next step, we analyzed the probability of progression-free survival at 10 years. We found a tendency toward increased PFS for KIR3DS1 positive individuals compared to KIR3DS1 negative patients. The HLA-BW4 had no significant impact on outcome. However, we noticed that among HLA-BW4 positive patients, the PFS tended to be improved in presence of KIR3DS1. The probability of PFS at 10 years was 77% for KIR3DS1 HLA-BW4 positive patients compared to 39% for KIR3DS1 negative. In the end, I would like to conclude that the presence of HLA-BW4, especially in coexistence of KIR3DS1 and KIR3DL1 genes, seems to protect against BCL. Moreover, the presence of KIR3DS1 gene together with HLA-BW4 gene is associated with longer progression-free survival. I would like to thank all the people who participate in this work, and of course all of you for your attention.
Thank you very much, Anna. Are there any questions, Campbell?
Thanks for a nice presentation. It raises a couple of questions in my mind. Do we know anything about loss of class 1 HLA expression on CLL cells?
It's downregulated.
It is downregulated. I had another question as well, but I've forgotten that one. One slight criticism. You've shown a nice difference in frequency of BW4 between controls and patients, and then you've looked at the combination of BW4 and 3DL1 or 3DS1, but you could also look at BW4 and blonde hair or black hair. Having shown that BW4 is decreased, whatever else you put in, BW4 will still be decreased. So you really need to take all those that have BW4 and then look at the frequency of 3DL1 within that group, or the frequency of 3DS1 within that group. Because if you're combining BW4 with something else, if BW4 itself is already decreased, a difference between the controls and patients, then whatever you combine with that will also be decreased.
Marcelo?
Yes. Did you check? I think there is a publication from Verheiden. Is that the Dutch? And Demane. Okay, I'm sorry. You're neighbors anyway. So I think they see the opposite. The homozygosity of BW4 associates with AML and CLL. Is that correct? Have you checked that?
Yes, I think so, but do you remember for how many patients?
No. However, I can tell you, we presented an abstract at APHIA and we confirmed the same thing. And there we had like 200 and something patients. So I'm checking my slides because I said this sounds familiar, but the opposite. But in a way, may have, I don't know, there may be many reasons for these opposite findings. Some of them could be genetic makeup and other factors that we may not be considering.
Yes. This might be a question for Campbell. With the Velarde study, the ALL wasn't killed by the KIA. So with the CLL, do we know who it is?
Personally, I don't know.
That was my question as well, actually.
But the other question was whether how homogeneous for this CLL was your group, because there are quite heterogeneity, I understand it, in the diagnosis with different mutations in different chromosomes. So we're all same type of CLL. I think that's something perhaps that in any study of disease association is quite critical, to try to work with a very as homogeneous as possible group from the many variables that compound the disease. Yeah. But the other question was, are these cells target for NK cells? Because certainly lymphoblasts don't seem to be.
So you understood the question?
Yes.
So people wonder whether these tumor cells are really targets for NK cells in order to explain the association you found. And that's certainly a point, although lysis might be one mechanism, but also it can be that NK cells do something in another way, like cytokine production or whatever.
Are they good targets? In which cases are they?
That's what she just asked, and it's not known whether this type of tumors are good targets.
There are few tumors which are missing all the HLA alleles, for example. These are exceptional cases, but I would suggest that these are the best targets. I don't know about others.
And maybe in addition to what Campbell was saying, if you do an analysis with different kind of markers, you can maybe also do, say, multifactorial analysis, whether some associations, what's the important association and what is primary and what's secondary. So that's also, but then probably you need a larger group of patients. Any more remarks? If not, thank you very much. So we go on with the next